This video is about genetic purity testing, seed technology part 2. Let us see what is genetic purity. Genetic purity is simply defined as true two-type plants or seeds conforming to the characteristics of the variety as described by the breeder. The principle of genetic purity is genetic purity or genuineness of the cultivar is tested by means of heritable character that may be morphological, physiological or chemical characteristics of seeds, seedlings or plants. The minimum standards for genetic purity for different classes of seeds is for breeder seed it is 100%, foundation 99, certified 98 and hybrid seeds 95%. The factors affecting genetic purity are natural crossing, mechanical admixture, random drift, mutation, selective influences of pests and diseases, etc. Coming to the criteria for Growout test to decide the genuineness of a variety. Growout test is mainly done to get the genetic purity confirmation. For maximum permissible off-type in the pure seed, the limit should be 0.10%. For then minimum genetic purity is 99.9% and number of plants required for the sample to be observed is 4000. Likewise, the off-type when it is 0.2, the genetic purity should be 99.8 and the samples drawn are 2000. When off-types are 0.3%, the genetic purity is 99.7% and samples 1350. When off-types are 0.5%, the purity is 99.5% and number of samples drawn are 800 or observation taken are 800. And when the off-types are 1 or more percent, the genetic purity minimum purity is 99 or below and the samples drawn are 400. There are two main approaches for genetic or varietal purity testing. First one is the use of computerized system to capture and process morphological information, and the second is use of biochemical methods to analyze various components of seeds, that is called as Kimo taxonomy. The two approaches of genetic or varietal purity testing are, one is machine version and another is Kimo taxonomy. Coming to the methods to assess genetic purity, there are three methods. One is morphological or we can say conventional Growout test. Then the second one is chemical and third is electrophoresis method. There are biochemical markers like protein or isozymes and molecular markers which are very convenient to assess the genetic purity with more assurance and more durability. Coming to the morphological test or conventional method, in laboratory, examination features of seeds like length, width, thickness, shape, weight, color, seed coat color, etc. and comparing them with those of authentic sample which are examined with naked eye or with magnified hand lens or with the help of scanning electron microscope. In field or greenhouse condition, the Growout test, how it is conducted. First, the seed sample is sown in the controlled condition with the authentic sample. Then genetic purity is determined on the basis of observation made on the plant morphological characters with reference to authentic sample. The genetic purity is always expressed in percentage. The size of submitted sample for Growout test is 1000 gram for maize, cotton, groundnut, soybean, species of other genera with seeds of similar size. That means always remember, the more weight of seeds is submitted for the the crops which are having grain size larger, or the seeds are quite having more weight than the other seeds. 500-gram samples is drawn for sorghum, wheat, paddy and species of other genera with seeds of similar size. 250 grams for beta species and species of other genera with seeds of similar size. 100 grams for bajra, jute, species of other genera. 250 tubers or plantlets or planting stakes, roots or cones are given for seed potato, sweet potato and other vegetatively propagated crops. Procedure for growout test. The first step is raising of desired population by following recommended cultural practices, example field preparation, size of the plot, etc. Then provide equal opportunity to each and every plant for full expression of genetically controlled characters. Sow the various samples of same variety or cultivar in succession and standard sample at suitable intervals. Adjusting the seed rate depending on germination percentage of individual samples and subsequent thinning is not required. This test is preferably conducted in areas to which the variety is recommended. Minimum 200 plants from controlled samples should be raised along with the test crop. The analyst employed for conducting growout test should possess the basic qualifications as identified under Seed Rules 1968. Here there are photographs representing the traditional approaches to purify testing. This is about the morphological test. The seeds based on their color, shape, size are characterized. Seedlings based on their length, vigor and other characters. In lab or greenhouse, the root characters and the shoot characters based on their morphology are characterized, and in field based on the color, shape and size of the different varieties are clearly identified. In the field condition, based on plant habit or fruit characters or flowering habit, the varietal identification or genetic purity can be assessed at morphological level. Coming to Kimo taxonomy, this is second kind. Kimo taxonomists means the person who are involved in this study. These Kimo taxonomists have recognized two groups of compounds that are generally useful in classification of plant species. The first one is episemantic or secondary metabolites like pigments or fatty acids, etc. Second one is cementioids or sense carrying molecules, may be proteins, nucleic acids, etc. The methods of this testing is based on analysis of secondary compounds, protein analysis and nucleic acid analysis. Coming to analysis of secondary compounds. This secondary compound analysis ranges from simple color test to complex chromatographic separations of phenols, anthocyanin, flavonoids and other compounds. Different tests include phenol test, peroxidase test, potassium hydroxide or bleach test, fluorescence test, hydrochloric HCl test, ferrous sulfate test, nitrogen oxide test, sodium hydroxide test, anthocyanin test and seedling pigmentation. This is a flowchart of phenol color test where different chemicals are used in order to assess the characters by getting different colors as end products. Coming to protein analysis. Because proteins are the direct gene product, the analysis of seedling protein and enzymes is most successful and widely used. Hence, much attention has been focused on seed storage proteins. There are two primary methods. One is various types of gel electrophoresis, and the second one is high pressure liquid chromatography. The first is with electrophoresis. Here, migration of the charged particles to medium may be agarose, polyacrylamide, starch, etc., under the influence of an electrical field. It is usually carried out in aqueous solution. A mixture of molecules of various sizes will migrate at different velocities and will be separated. The varieties are verified on the basis of banding pattern by measuring the specificity of bands, total number of bands, presence or absence of specific bands, or intensity of band. Coming to gel electrophoresis based type of separation. Here there are two things. Native one, that is separation by size and charge, that is measured by charge by mass ratio. Then denaturing, that is separation by size, and others like two-dimensional electrophoresis or two-dimensional electrophoresis. Gel electrophoresis in the native continuous system, gel and tank buffers are the same single phase gel, example page, agarose and starch gels. In discontinuous system, gel and a tank buffers are different, that is two phase gel or stacking gel like page. Gel electrophoresis based on denaturation, SDS, that is sodium dodecyl sulfate, used to denature protein, that is a discontinuous system. Here urea or former amide is used to denature DNA or RNA. Other types are, first one is isoelectric focusing, that is protein separation based on isoelectric point in the pH gradient, and the another one is 2D electrophoresis, that is combination of IEF and SDS page. Here the pictorial representation of steps of isoelectric focusing is given. First step includes crushed seeds are assessed subjected for protein extraction, then vertexing by adding the extraction solution, loading of samples to gel, then gel running, taking off the gel once done, then staining the gel. After staining, destaining the gel, blotting the stained gel, and gel is ready to read. This is an old technique, but nowadays many of the advanced techniques are coming where the steps followed are very less and within an hour the results are obtained. This is DNA test for hybrid seed purity assessment. The procedure follows. First, the 2 to 3 day old seedlings are taken and then tease out the shoot and root using sterile forceps. Then microcentrifuge tubes with 200 microliters of extraction buffer. Subject it to motorized pestle for grinding. Then treat it with 100 degrees Celsius for 10 minutes. Then centrifuge for 10,000 for 2 minutes, 10,000 rpm. Then use the supernatant at template DNA for PCR. Then subject to PCR, run the gel and get the image. Finally score the bands. This is for DNA based method. This is a simplified version of the previous slide where just amplification, electrophoresis and detection are the shortest steps. The benefits of using molecular markers: the detection of variation directly at DNA level, highly abundant and polymorphic, independent from environment and reproducible, expressed in all developmental stages, known position in the genome, and automation is possible here. This is about the PCR reaction. I need not to tell you. If you are not thorough, just read out the procedure. The steps are given here. Coming to advantages of assessing the genetic purity. Genetic purity is helpful in plant varietal protection, registration, certification, and patenting. Assurance of genetic purity for ensuring better agronomic performance and predicted expectations. Prevention of misappropriation and willful admixture of seeds or cultivars at commercial or farmers level. To detect even the minute genetic differences between cultivars very uh, appropriately for existence of novelty among essentially derived varieties. Genetic purity analysis is important factor for quality seed. For the farmers, it helps farmers how like no loss because of poor seeds plus higher returns. For producers, marketing grip, they can assure the farmers about their quality, and technologies in hand means used for the benefit of humankind. Thank you. I hope you liked this video and don't forget to subscribe my channel. Thanks once again.